Thank you to everyone. Thank you for dialing in for the this episode of the 2024 State of Play series. And for those of you on Zoom, you may see a notification about the meeting being recorded. So please just click OK or agree to continue. So firstly, an acknowledgement of country. MND Australia acknowledges the traditional custodians of country throughout Australia and the connections to land, sea and community, and we pay our respect to the elders past, present and future, and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respect to elders past, present and emerging, and extend a welcome to any Aboriginal and Torres Strait Islanders peoples on this call. So tonight we've got a different format to previously. We're going straight into a Q&A. We have 4 fantastic experts from right across the MND research spectrum, and I'll let them introduce themselves in a minute. So each presenter will just let them give a chance to give a quick overview of their research and expertise, and then we'll go straight into the questions. We've had a great response to our request for questions, so there are quite a few lined up, but if you have more questions, please fire in more through the Q&A function in Zoom, or if you're on Facebook, then you can add your comments, your questions in the comments, and we will collate them and get them into our Q&A tonight. And hopefully we'll get through them all, and if we don't, then we'll probably put another one of these on to make sure we can answer all your questions. So I think we'll start off with Kelly's going first to introduce herself, and then we'll go through the rest of the panel. So thank you. Take it away, Kelly.
No, thank you, Gethin. So I'm Associate Professor Kelly Williams, Head of Genomics and Bioinformatics at Macquarie University MND Research Centre. So it's a large concentrated research centre with around 70 staff and students that are dedicated to MND research. So I'm currently a FIGHT MND Mid-Career Research Fellow. I was a former MND-RA Early Career Research Fellow. I've been working in the field of MND genetics for the past 17 years. So when I first began, only one gene was known to cause MND, and now we have around 30 to 40 genes linked to MND. And I'm very proud to say our Australian genetics research program has been involved in a large number of these discoveries. So at Macquarie, mine and my research teams, we focus on both the familial, so the inherited and the sporadic form of MND, and we perform small scale through to very large scale genomic studies where we look at the entire genetic code, so the entire human genome of people living with MND. And we're trying to simply find genetic links to disease to help us understand better towards treatments.
Great, thanks, Kelly. I'll go next. So I'm Associate Professor Adam Walker. I'm the lab head of the Neurodegeneration Pathobiology Lab, which is based in the Clem Jones Centre for Ageing Dementia Research at the Queensland Brain Institute of the University of Queensland. I'm also a FIGHT MND Mid-Career Research Fellow, and I'm also the Ross McLean Fellow at QBI, the Queensland Brain Institute. My lab studies the molecular mechanisms of motor neuron disease. So I have a PhD in the cell biology of MND, and I've undertaken research employing both cell biology and also genetically modified mouse models of disease. My lab aims to understand the processes involved in protein aggregation and cell stress responses and other biochemical changes within neurons that lead to neurodegeneration in motor neuron disease, with the goal of identifying better and new therapeutic targets for MND.
Hello, I'm Karen, Dr. Karen Hutchinson. I'm a research fellow with the Australian Institute of Health Innovation at Macquarie University, and I'm also an honorary conjoint scholar with the Central Coast Local Health District. And I'm also a practicing physiotherapist as well. So my sort of research area is exploring experiences of individuals and family members living with complex neurological conditions. Also looking at co-design, implementing and evaluating different models of integrated care into practice. So I've been evaluating a motor neuron disease clinic that we set up in the Central Coast. We've been sort of looking at how that some of the barriers and facilitators in that clinic. I'm also a physiotherapist, and I've worked for many years with people living with MND in the UK and in Australia. And thank you for having me today.
So thank you, all of you. And my name is Parvathi Menon. I'm a neurologist, associate professor with the University of Sydney. So I'm a clinician. My primary job is to diagnose people with MND, which is not always very pleasant. And then the second thing I do is I manage them through the MND clinic at Concord Hospital, where I'm a staff neurologist. The other thing we do is also participate in therapeutic trials in MND, so that patients have a chance to try out whatever new medications are available. My area of research is into the neurophysiology of the brain and looking at changes that happen in the brain with motor neuron disease. And we've published a number of studies in patients saying there is a phenomenon of cortical hyperexcitability that happens sometimes even before the onset of disease in people carrying gene mutations. And that's an area which we are following through with a lot more research. So I'll stop there, Kethan, and ask questions.
Yep, thank you, Parvathi. And as I'm sure you can now see that we've got a fantastic range of knowledge and skills across MND here tonight. So I'll start going through some of the questions and let's see where we go. So first question, let's have two new therapies have emerged in Australia recently, Tofersen is now available for those with the SOD1 mutation. Has this been trialled in people with sporadic MND? And also then, what about edavarone as well? Is that expected to be, when is it going to be available in Australia? And could we just talk about why it takes so long to get some of these treatments in Australia when we see them overseas first?
So
do you want to talk to edavarone perhaps first, Parvathi?
Yes, I will. And so edavarone, as people know, has been approved by the FDA, but the European arm and the European Association has said that has not been effective in large clinical trials that they've conducted. In Australia, it's actually approved by TGA for use in motor neuron disease. So people who wish to procure edavarone can do so. And it's just about having a center which is able to infuse it. So it's the IV form of edavarone, which has been approved by TGA. And where approval by PBS is concerned, I think applications have been made by the pharmaceutical company, which is going to process it in Australia. But I think this is still pending, and it's all about funding and costing to see if the PBS feels that it's going to be valuable to use in Australia. So those who are interested in using edavarone can still procure the medications from overseas and look at a center that's their local MND clinic to see if they can get the infusions at their center.
Yeah, I think Teva, the company, bringing it in, and I think it's been nominally approved by the PBS, but they're still negotiating the final price. So I think they're looking at early next year, hopefully, to have it available. Kelly, would you like to speak to the idea about testing Tofersen in sporadic patients?
Yeah, so Tofersen, unfortunately, is not for sporadic MND, as it specifically is to target a defective SOD1 gene. So for those people living with MND that have a SOD mutation, or perhaps younger members of their family who have a SOD mutation but have not yet had disease, which is for a pre-symptomatic clinical trial. So Tofersen specifically targets defective SOD1. So the only way it would be useful in sporadic MND is if they actually have had genetic testing and have shown that they have a specific mutation in SOD1. However, when we did a genetic ancestry study a couple of years ago, looking at I think around 90 familial MND patients with SOD and SOD1 mutation, and 3 sporadics that we had through our clinic and through historical DNA collections. And we found that those 3 apparently sporadic SOD1 carriers were in fact distant relatives of these families. So it's unlikely to ever see a true sporadic case that has a SOD mutation where a tofersen would be effective.
Okay. So another question here is Adam can lead off on this one, perhaps. So if the main problem with ALS is protein aggregation, what are the benefits of an autophagic drug like monipantel that has shown some benefits in their early stage 1 trial? And are there other sort of treatments looking at this protein aggregation?
Yeah, so the process of protein aggregation is really one of the key hypotheses of what is causing and driving neurons to degenerate in motor neuron disease. So in almost all people who have motor neuron disease, we know the protein, it's called TDP43, which aggregates inside the neurons in the brain and the spinal cord of people with MND. And the clearance of these protein aggregates has been shown to be beneficial in cell and animal models of motor neuron disease. So that is definitely sort of a key question in the field. So there are multiple autophagy-related drugs that stimulate the autophagic protein clearance pathway. So this is a biochemical pathway that is involved in getting rid of these aberrant protein aggregates within neurons. And we know that that pathway, it's normally active in all neurons, but particularly in neurons that have these aggregates, its stimulation can lead to faster clearance of the aggregates. So drugs such as monipantel and others are certainly active areas of research for MND. I think it's an interesting and promising area. One of the issues with this pathway is because it's so important in all cells, it's not just in motor neurons and not just in all neurons. It's really important that we have the right way to trigger the pathway to stimulate increased clearance of proteins without having off-target and sort of side effects. So that's a really important part of the studies looking at these pathways.
Thank you, Adam. A question for Karen here. Can exercise help to slow down symptoms? And if yes, what type of exercise is best?
Yes, there's been quite a lot of different reviews related to the benefits of exercise and what types of exercise. And most of the data that comes, the evidence that comes out on this is really, moderate exercise is the most important sort of things to consider. There's been trials where aerobic exercise, which is where you're working on your cardiovascular fitness and some strength-based, but it really isn't about maximizing all the strength, because your fatigue is a huge issue with over-exercising, which can actually be detrimental to any benefits that you're getting from the exercise. But what they're finding is that the short-term gains are often not seen. It's a bit longer. From about 3 months, you might see some benefits. The type of exercises that people tend to do, they've talked about doing exercise every day, or 2 or 3 times a day, and sometimes 2-3 times a week, sorry. And so that's actually beneficial than actually over-exercising with MND. And so some people have found that swimming can actually be quite good for those that are still able to get into the pool. And just not doing more exercise than like 60 minutes a day is detrimental again to the muscle activity. Sometimes some of the benefits that you can get from reducing pain, reducing sort of that contractures that you can get in joints, improve posture. So there's other effects of the exercise that can be beneficial. And depending on, there isn't really specific you'd have to be assessed by a physiotherapist to see which is the best type of exercises that you do. But it actually does help to reduce some of that deconditioning that you can get if you're sort of sitting and not exercising much at all. And it can be translated into being able to walk maybe longer, being able to get out of the chair, being able to roll over, but in bed. But it again, it has to be very much assessed by the physio that you're seeing.
Thank you, Karen. A question, I think for Kelly, next one. Is there any research or clinical trials underway to look at C9orf72?
Okay, so C9orf72 is unfortunately quite a it's quite a complex genetic mutation. So when we think of our genetic code, it's like 3 billion letters of DNA, and in people living with MND that have a mutation, a genetic mutation in SOD1, it's a single change at one of those 3 billion DNA units. But C9orf72 is different, where we are looking at what's called a short tandem repeat in the genome. So rather than being a single DNA base or a single unit of DNA code, there's actually there's 6 units of DNA code. And in, I guess, the normal population, most people would have 3 to 23 repeats. So it's quite a small area of your genome. However, C9orf72 repeat expansion, these 6 nuclear or 6 DNA unit repeats can actually be expanded up to 1,000 times. So when gene therapies that are aiming to target defective genes are almost looking to, I guess, if you think about, if we're thinking about a linear genome, they're almost trying to sit on top of a gene defect to block it. But because C9orf72 is so big, it's very complex. It also is complex when building models, so mouse models, cell models, zebrafish models. Because of the unique type of genetic mutation that it is, it makes it very difficult. So it's people desperately want to be able to develop a gene therapy for C9orf72 because it is the most common genetic form of MND. So it's complex. It's complex, and that's why we're not, I guess, seeing groundbreaking breakthroughs in that field.
Yeah, unfortunately, there's been 2 trials that have looked at it, and both of those have failed. So I think everything's gone a step backwards, unfortunately. While I think everyone's looking for a better way to look at that gene. A question for Parvathi here. Is denial a common response after an MND diagnosis, and how should family members deal with this kind of response?
Yeah, it's a difficult problem, isn't it? So I think denial is a feature in a number of severe or significant diagnoses, like with a number of malignancies and other diseases, as with MND, when you know the outlook is not good and there are not many chances of treatment and cure, people do go into denial. So it can be very difficult. In my practice, what I try and do, and I do meet some people with a very prolonged course who still haven't accepted their diagnosis. And I think the reasonable way to handle the problem is to give them evidence, not just brush aside their concerns, but to provide them with evidence to say why we think the diagnosis is correct and other diagnoses that they raise are not sustainable. So if it requires a certain degree of investigation to be completed, I do it because I think it's a reasonable request. And when it comes to the denial itself affecting their treatment, well, there's not a lot you can do because you can't compel a person to accept a diagnosis or to choose a treatment. They can deny it. And while it's difficult for the family, what I would say is we can support the individual with the problem. The family might struggle. I think you need to look at your own mental health and see where you can get help from your GP. But the primary person at the center of all of this, I think, is the patient. And what I think we need to do is to support them through that journey of understanding their condition, accepting their diagnosis, accepting treatment, and if they have valid concerns, trying to answer it and investigating further as required. I think that's a reasonable way to approach the problem, because this is not uncommon, not just in MND, but in other severe or neurodegenerative conditions. Like, people will not accept their diagnosis, and they may seek second opinions, third opinions, as many as required. And I try and facilitate that. So yes, seek another opinion, get more opinions, and complete investigations if there are any concerns. But at the end of the day, what we have to do is provide them with evidence, which is like factual. You cannot hide that. And so that they can, and then provide them with avenues of what can be done, like options that are available by way of management, rather than just closing it at a diagnosis.
Karen, did you want to add anything to that or not, from your experience?
I mean, the only thing I can sort of add to that as well is that is the family being supported in managing that as well. And often, you know, there are young people in the family too. So just making sure that, you know, everybody understands. So the importance of carers and being able to manage that situation is really important.
Thank you. Question probably for Adam. Is anyone aware of immunotherapy or cell-based regenerative trials of medicine that are currently underway?
Yeah, good question. So there are lots of areas of research involving the immune system. There is a large clinical trial at the moment going on with interleukin-2, which is aimed at modulating part of the inflammatory related pathways in motor neuron disease. In terms of cell regenerative therapies, that has been quite controversial in the MND space. There have been some clinical trials and not a lot of evidence to show that they're necessarily beneficial, but research still ongoing. I'm not sure if Parvathi, you might want to comment on those as well.
Yes, I'm not aware of any that are currently happening in Australia. So we have completed trials, including the ones about which people had concerns, the TEAL study, which used Tecfidera, an agent which modifies the level of regulatory T cells. But so far, I don't think we've had trials which have reported significant benefit in their primary endpoints with all of the immunomodulatory therapies which have been used. And that's the unfortunate part.
Right. Thank you. Next question. So this is a question for Kelly. What is the most effective way that a family with a MND familial link can contribute to research or finding a cure for MND?
My apologies. When we have MND families present at the clinic, they'll fall into one of two categories. So category one is where we either know the gene mutation that causes motor neuron disease in their family, or we find it through diagnostic screening. So this is around 2/3 of MND families. So they'll have a genetic mutation in a common gene that we know to definitively cause motor neuron disease. And then the other category is accounts for 1/3 of MND families where they do not have a known gene mutation, and we cannot find one. And so the best way, I guess, across either category is how people with a familial link can help research is, I guess, number one is to donate samples, so biospecimens. So for example, if we at our neurology clinic at Macquarie, we have an associated biobank. So we consent families, so MND families, so people living with MND, spouse, any related family member, we can send them into our biobank, and they will donate blood, which we get DNA, RNA, serum, plasma. And so that's, by donating samples, it allows us to do something. It allows us to conduct a whole lot of different research, whether they have a no mutation or not. Also, from a genealogy perspective, if families are able to actually generate large family trees, or if they can track their genealogy to be able to provide to the clinic, it gives it can give insights for, say, the genetic counsellor to see what the penetrance of disease is. So whether it's if it's an inherited form, a motor neuron disease, if we're seeing 50% of people in a single generation becoming affected, which is what you statistically would expect in an autosomal dominant disease, sorry. So having these large families is really helpful for research. It also allows us, from a genealogy perspective, to link different branches of the family, which can, if we have to do genetic studies, particularly for families where we don't know the gene mutation, the more DNA samples that we can get from people living with MND, the more statistically powerful it is to try to find new gene mutations. If someone has a known form of the disease, so if they have a mutation, say in SOD1 or C9orf72, biospecimens is still just as important, because we see in these large families, we see a lot of variability in, say, age of disease onset or progression. You know, sometimes someone might become affected at 20, and someone with the exact same mutation in the same family might become affected at 90. So when we see this, it suggests that there might be other genetic modifiers. There is something modifying disease, and it could be genetic risk factors. So if we have large families, lots of, say, DNA samples or RNA samples, we can look for these modifying factors, which also, I guess, you know, something that we hope that we could find out of modifier studies is if something is causing either a longer disease progression, so allowing people to live longer after they become affected, or not actually developing MND until they're elderly, potentially that's an avenue for treatment, finding something to modify disease. So that's how, yeah, they can help. Family trees, biospecimens.
Thank you, Kelly. And just out of interest, if people aren't going to the Macquarie Clinic, can they still contribute their samples from other parts of the country?
This is another complex question. At Macquarie, we do remote collections for families, specifically for MND families, because we know that we know that we can do genetic testing. The neurologist, our new head neurologist, Professor Dominic Rowe, always, if we have people donate samples to the biobank, he always wants to have diagnosed them. So we're clear that we're actually people living with MND in the biobank, maybe not some other form of neurodegenerative disease. I would like to see that happen, where we can get samples from all around Australia. And I think that might also, I guess, link into, you know, finding mutation hotspots as well. Perhaps we'll get in touch with MNDRA. We see what we can do if there's people, particularly if they're families, to do remote collections. So.
Right. Thank you.
And I'd like to add in that, Gethin, that we've also undertaken like the change that happens in the brain. So it's called hyperexcitability. And the thing we found is that no matter what form of MND you have, whether it's genetic or sporadic, it seems to be like a pre-terminal change that happens in the brain. And we have followed families up with known gene mutations and have detected the change of hyperexcitability just before they develop clinical symptoms. And sometimes we follow them up for a while. And now there are additional markers available, which tell us about neuronal loss and neuronal changes. So this is a project we also undertake. And sometimes it's the family members don't need to know whether they have a mutation or not, though we as the researchers might know. So it can be done either ways. And looking at a number of markers of what changes, when it changes, can we predict? and can we intervene in that is obviously the effort that we are doing.
Next question, probably for Karen and Parvathi as well. Is there any information on alternative therapies or on the use of substances, things like metformin, creatine, bicalin, acupuncture, cannabis oil? Do you have any feel for any of these all sort of alternative therapies provide any benefit? Or what does work?
Yeah, look, I might start with that because this, in fact, the questions that came through before this session made me go and look at a number of these alternate therapies. And obviously they haven't been effective. And a site which I found useful and which the MND Association itself recommends is ALS Untangled, who are actually curating what you call, it's maybe over-the-counter medications, other preparations, herbal medications, acupuncture, all of that. And they actually list each of these interventions and what's the information available on it and what's the advice on it. And I thought that was a well-curated site because these therapies, some of them have been tried, like cannabis is currently being tried at the Gold Coast as a therapeutic trial, and people who are interested and can get to it could register their interest. But till these trials are done, and some of the trials have been negative. So what's the merit in continuing them? But some of these medications are like, they're not medications, they're just supplements and such. And so is there any harm in trying them? That's the question that people would ask. It's a very complex area because there are so many products available. And I think that site, ALS Untangled, which has been mentioned on the MND Association site, is actually a very good site to look at what's available and what's the evidence on it. What do they recommend at the end of having assessed all the data that's available?
Karen, did you want to add anything?
Yeah. I mean, just from a clinical perspective, as a physio, we get lots of we have lots of conversations about trialing different types of therapies, and definitely cannabis oil is I think is a common one. And I know from my experience as a physio that it has been used by a large number of people with MND. And that's just from my clinical experience. And some people feel that it's really beneficial. Some are not quite so sure. So, you know, like as Parvathi said, it can vary with different people, and a lot of people will experiment a bit with different types of things. Acupuncture is definitely another one. You know, homeopathy, you know, all of those type of different types of therapies people have trialed. But I think it sort of comes down to the individual, but as you know, like the evidence there is limited at the moment.
Thank you, Karen. A question for Adam here. We see a lot of failures with mouse studies, certainly in their translation through to humans. So why are we sort of keeping going with mouse studies? Are there better options?
Yeah, it's certainly true that there have been a lot of studies in mice where drugs or other therapies have shown some benefit that then has certainly not translated to human trials. I think there are a few different reasons for that. One is that quite a few of these mouse studies were not done in a particularly well designed manner, and so you may get what looks like some great evidence from a mouse study, but when you look at the data, you realize that there were only a small number of animals tested, or the readouts were not very clear, or the actual tests that were used in those animals may not directly translate to what we can look at in people. So I think it is important that trials that are done in animal models, in mouse models, for example, are done better than they have been a lot in the past. But in addition, mice are not just little people, they are a completely different species. So there are certainly mechanisms, biochemical pathways that occur in human motor neuron disease that are not found in mice or other animals. So I think that one thing that we need to keep in mind in our studies in model systems is that we need to be clear about what the scientific question is that we're asking. We're not trying to cure our mouse or our fish or our rat or whatever it is that we're testing. We're trying to use that as a way to answer a scientific question. So if we want to look at the autophagic protein degradation pathway, then we need to design our experiments in these model systems in a really clear, well-controlled scientific manner to get an answer that we can then apply to understanding human disease. But in addition to testing in rodents and other species, there's a lot more research in the last few years, and it's accelerating, and a lot of labs around Australia and around the world are now using human stem cells that are reprogrammed from the skin or other cell types from people who have motor neuron disease or have died with motor neuron disease. And these are certainly a really great model to use in the lab. So that allows us to look at human neurons or other cell types that are involved in disease in a dish in the lab. I think that a lot of labs are using these now, and that will allow us to look at some of these biochemical pathways that are only found in humans and not in other species. But I don't think that there is, I don't think we're at the point where we can move away from doing animal testing for diseases like motor neuron disease yet, because just studying cells in a dish or neurons grown in tissue culture doesn't allow us to look at behavioral readouts, for example, which is a great way that we can use mouse and other animal systems to test drugs and therapies that have a readout in terms of improving motor function and motor decline, which is not really possible in our cell and neuron systems. And I think it's just important that we keep in mind that the experiments are designed very carefully and the interpretations are very careful as well, rather than jumping into conclusions.
Great. Thank you, Adam. The question is a more general one. Several of you might have a comment on this. So is generative AI being used, or could it be used to advance MND research or improve the quality of life care for people living with MND? So just wondering if you've got some good examples about where it's being used. I think you probably all have. Anyway, I'll let you run with that one.
Maybe I'll go first. So in terms of AI, certainly there are so many possibilities of what we could use AI for. In my lab, we're not necessarily using it every day on the day-to-day in our research, but one example in my field is looking at protein aggregation and protein function and protein structure. Google have now released a program called AlphaFold, which can predict the structure of any protein. So there are hundreds of thousands of proteins in the human body and millions known across different species. And until a year or two or three ago, we only knew the structure and how these proteins are folded for maybe 100,000 of them. So Google have used AI to allow us to now predict the structure and the folding of every single protein that is known to humankind so far. And so that is a way that we can even more quickly sort of look at the structure of proteins and design drugs to bind to them. And certainly that's something that is being done a lot more in all biomedical research, including in motor neuron disease. Any other examples from anyone? How about in the genetic space, Kelly?
Generative AI, no. So I guess from an AI perspective, we're more likely to use machine learning, looking for patterns, looking through clinical data sets, genetic data sets, matched RNA, so transcriptomic data sets. For us, the main way in which we're using it is to try to pull out different clusters of people living with MND, because it is such a heterogeneous disease, and that's probably why there might not be one perfect treatment for MND. So by trying to pull out these different clusters, using machine learning, using all of the data sets that we've got effectively, if we can find these different clusters of patients, and maybe they have specific clinical features that are common amongst them. This stratification may enable I guess it's the first step towards personalized medicine. So stratification, if we find that there might be, I can't even think of a good example, perhaps tall males, for lack of a better example, there may be some specific treatment that just works for them better. So any other person that presents with MND that's not a tall male, it may not be useful, but we know straight away that this cluster of people is very suitable for this specific treatment. So that's patient stratification through machine learning.
And I think perhaps some of the other earlier examples, maybe not AI, but as you say, machine learning is things like text, predictive text in eye gaze, etc., like that, and text to speech. I think they're probably the very early examples where this kind of technology was being used. Next question. Why is there a 2-year post-diagnosis cutoff for a lot of clinical trial participation? And could this be reconsidered for people with more slowly progressing forms of the disease?
So I could pick that up, Kevin. So, well, it's what we're trying to do when we design a clinical trial is to try and get all people in both the treatment and the placebo arm to be comparable. And what we understand about the pathogenesis of MND is that there is the preclinical phase. There are things happening before there is motor neuron loss evident as disease. So most of the intervention that is planned is with the plan of reversing that motor neuron loss. And so if you want to do that, you really want to do that early in the disease process. That perhaps is one of the reasons why we try and target people who are in that early phase of their disease, and so that they're comparable, have had equal amount of symptom duration, and then you randomize them into groups of those who get the treatment and those who get the placebo, see if there's a big difference from the intervention that you're trying. What if you expanded that to include people who had disease for 2 years? Well, the common form of MND, which is the ALS with rapid progression, people would have significant disability by that stage, and their focus would not be on therapeutic trials. So you might not be able to recruit people at that duration. And then you would have to expand the cohort to include a large number of people who are of that same duration, so that you can balance it out. And that might make the trial more difficult to conclude. That's the only reason why, because most trials are not run by large pharmaceutical companies. They're run by small groups who have small amounts of funding. And what they're trying to look at is like, will an intervention work? So who shall we recruit? People who are really early, don't have a lot of motor neuron loss. Can I arrest that process by my intervention and then see what happens in the one arm that gets the intervention and the other which doesn't? And I think that's the reason why study trial designs are made in such a way, at least for the therapeutic trials. There are a lot of other trials in MND which, looking at things like hyperexcitability, other databases, etc., which do not have a timeline of 2 years. And so it's not to try and disadvantage patients. If there was any benefit from the treatment, I think it would then generalize for everybody. That's the idea behind concluding the trial rapidly within a few months and having a decision, deciding whether you're going to move on and have a further phase of the study and get benefit out to the most number of people as early as possible. I wonder whether anybody else has any comments on this.
Okay. Next question is it's probably sitting in Karen's basket a bit. Is like people are concerned that they don't hear much about the follow-ups after they are engaging in research. How can they always make sure they stay up to date what about the outcomes from their research, and where can they find out the information rather than just reading about the next breakthrough perhaps in the media?
I think that's quite a common question, and I know both as a researcher and as a clinician that we have many discussions about that, about, you know, how do I get to know the outcome of the, you know, if it's a clinical trial or some of the other research that sort of I'm involved in is more actual in the healthcare space. So it's actually care delivery. And I know when you usually consent to those to you know, participating in the research, is that there may be a clause where they ask, do you want to hear about, you know, the outcomes of the research? And I think it's really important to make sure that if you are interested in that, that you let people know that you want to be informed the outcomes. Because I know that uncertainty has, you know, can be quite distressing for people when they're not sure, especially with some of the clinical trials. But I know from my experience, I do let people know the outcomes, and we've actually run in the research that I do, we have like advisory groups, and actually we have people that are participating in the research that sort of contribute to the design and the implementation of the research, and sort of participate all the way through. So that way they are kept up to date as to what is happening in that type of research. But I know with clinical trials, that's that may be different.
Thank you. Anyone else perhaps want to comment on what research is being conducted to understand hotspots of MND currently? I think this might be in your basket, Kelly.
Yes, so hotspots, one of the key, I guess, problems for finding hotspots is that we do not know of every single MND diagnosis in Australia. And so our head neurologist at Macquarie, Professor Dominic Rowe, this is his, I guess his most hot topic at the moment is petitioning the government to make MND a notifiable disease, because without actually knowing where every single patient is, we can't accurately find hotspots. And there can be, I guess, sometimes rumours of hotspots that may come up, and it can be due to large MND families. So it may not actually be a true hotspot. It may be there is just a large MND family that, you know, has many offspring that live in that one particular area, which inflates the results. But I'm, I guess, excited to say last month it was announced that we received funding from New South Wales Health to specifically start to look at hotspots in New South Wales. So we will be using like geolocation data and seeing if we are actually able to use patients that we have collected at Macquarie over the last, I think, 15 years, see if there are any regions in New South Wales that seem to have a higher incidence of MND with emitting families from that study, and then looking at the biospecimens that we've collected from these people living with MND to see if we can find any potential environmental clues, factors, triggers in the biospecimens that we have collected. So it's, you know, as I said, I guess the key point is without knowing where every single patient is, we can't accurately define hotspots.
Yes, and certainly data collection is becoming a really key issue, and MND Australia are working as well with a data strategy to try and improve our data collection right across Australia. I mean, ideally, yes, every patient gets logged as MND, and we know where and who they are, and so that's what we're building towards.
And
which is amazing.
Yeah, can I just add to that? I mean, we have issues around you know, sharing of medical records, that type of you know, all that data. It is very complex, and I noticed you said New South Wales, because then once you start moving into other states and territories, there's, you know, all the other complexity. And that's the same that people, you know, the issues people have getting care and support is navigating all those different, like having shared medical records, being able to communicate much more effectively. And so I feel that if we could have some improvement within New South Wales, just in this you know, example, that would be a great way of getting some more data if we could actually, you know, share and collaborate all across all the different you know, primary hospital, you know, community care. I think that would be a great way of gathering that data. But I do hear a lot about little hotspots across Australia that people talk about. Yeah. Thank you.
We have a couple of questions here around the Healy platform. People may be aware that the Moni-Pantel trial for its phase 2 will be going to the Healy platform trial in the US. I wonder if anyone's got any thoughts on the benefits of the platform trial approach, and 2, what are the chances of us establishing a platform trial in Australia? I don't know, perhaps Parvathi, can you discuss this?
Yeah, so I guess the advantage of the platform trial when it was designed itself was to speed up trialing multiple agents without having to wait for one trial to conclude. So that's the advantage. But obviously, it's been supported by that enormous fund which was received by the group at Mass General to set up the platform. And we need to start thinking about something similar, and researchers in the space are trying it. It would just, you know, like have decisions on whether a therapeutic agent is worth pursuing further or not. And you don't have to wait for one trial to conclude before you start another. You can have multiple arms running simultaneously. And that's the advantage of the platform trial. And so definitely something for us to aspire towards. It's a thought about how researchers are going to get that kind of funding and how we're going to organize it. If it can happen in Australia, that would be good.
Thank you, Parvathi. Sorry, I'm scrolling back up here. We've had a couple of questions about genetic testing as well. So less about specific research. It's like, can genetic testing be taken anonymously without the person having the test being told? And what is the best way to access genetic testing and counselling, especially perhaps if you live in a remote location? And then there's another question is, can you actually test people post-mortem? So can you take a sample from someone perhaps before they die, and then if they didn't want to be tested, you can then find out that information later for whatever reason you need to do that? Sorry, there's about 6 questions there, Kelly. But...
That's okay. I might start with the post-mortem one, so that yes, you can, because you can give next of kin consent after the person has passed away. So yes, if there is a sample taken beforehand, it can be tested. However, I guess genetic testing, when new patients come into the clinic, every single person that come into our clinic gets a genetic panel done to see, specifically to see if they have a mutation in SOD1, so they're able to go onto tofersen. But it also allows us to maybe find rarer genetic forms of disease. But in terms of, I guess, genetic testing, if people don't want to know, unless there is a family history of disease and a known gene mutation, it's actually not going to give any information. And it's sometimes we get this question in relation to pre-implantation genetic diagnosis and IVF. So someone may have a family history of motor neuron disease. They know they're at risk because they've had a parent that has had motor neuron disease, and they would like to undergo pre-implantation genetic diagnosis and IVF to avoid potentially passing on the mutation, but they don't want to know themselves if they actually have it. And that is possible to do blinded IVF. However, with any, I guess, any dealings with a genetic form of disease or genetic testing, particularly if there is a family history and a known genetic mutation, it's so important for people to go through genetic counselling, because whether it's next of kin testing, next of kin consent for testing, or whether it's someone that has a family history that they want to find out if they potentially carry a mutation, they need to go see a genetic counsellor. And I believe it actually might be mandatory, because individuals need to know pros and cons about finding out their actual genetic result. In terms of I guess another thing about, say a sample is donated for research, and part of our, I guess, standard research is that we do genetic testing. When a person signs a consent form, they can, there's normally a checkbox in there, like, can you return results to me, or I do not want to know results. And so that's also, I guess, a personal decision of from a research perspective. So I hope I've answered all of the questions.
Yeah, just a quick extra one. Is there a service? Can you get genetic counselling by telehealth? Do you know if that's an option at the moment, especially for those people who aren't living in well-serviced areas?
I don't actually know. I imagine the best option would be to speak to the neurologist that has seen you and diagnosed you, and see if they can refer you to a genetic counsellor, because in my opinion, I think it's important to see a genetic counsellor that has experience in complex diseases, because Mendelian genetic diseases are very different to receive genetic counselling for something that's slightly more complex, such as motor neuron disease, where, as I mentioned before, we can have reduced penetrance in a family where a person might not become affected till they're quite old. Yes, my recommendation would be to speak to your neurologist and see if they can refer you to someone.
I might just add there, Ketan, that's true. So I think the first port of call is the diagnosing neurologist. So it might not be for the patient alone, but for the family members. And the good news is that genetic testing has become, especially in symptomatic people, has become Medicare rebated now. So that's very helpful. And a genetic counseling generally goes via your health district. So you don't need to be anywhere near the neurologist, but where you live, the neurologist can guide your GP to refer you on to a particular group, who then will counsel you before you undertake testing. So for the Medicare rebate is mainly for the predictive, rather than for, not for the predictive, for the patients rather than for family members. I'm not sure that carrier testing is available except for pre-implantation, maybe.
Thank you, Parvathi. We've had a couple of questions here also about studies on people with slower progressing or who have lived longer with MND. I don't know if anyone here has some thoughts or insights into those studies. I know there's some being done. And, can anyone talk about some of the studies being done either here or overseas?
I could just add into that, like the forms which are truly slow progressing are commonly PLS, which is not even thought to be a pure form of ALS. It might be a different variant. And the other form is subtypes of ALS, which, like the flail arm or the flail-like ALS, which are just very slow to progress. Now, in PLS, I'm not aware of any trials that are currently running in Australia, but we do undertake studies on their cortex to see if there's any changes we can record. And commonly, there is no recording possible because of a phenomenon called inexcitability in patients with PLS and such forms of ALS. Now, the flail arm syndromes, there are no trials running, but there are very clear markers of higher risk in some groups of the slowly progressive variants. And these are well recognized now by ALS clinicians. So they're looking for those problems. And so like the flail arm ALS, if there's respiratory involvement, it's unlikely to progress as a flail arm variant alone, but is likely to have more disability early. So where trials are concerned, I'm not aware of any that are running here. And I've looking at the international trial registry, I'm not aware of any trials specifically for slowly progressive forms. But there are some particular characteristics of these variants which are useful to discuss with your clinician.
Yeah, I don't know about any trials either, but I know there's a study going on at University of Queensland, actually, Tom Shaw, over at the National Imaging Centre. And I think he's using very high resolution MRI to look at patients. Do you know anything more about that one, Adam?
I don't know anything about the outcomes, but yes, I do know that they're using very fancy MRI that can detect really small changes. So some interesting research being done there.
Yeah, so that's a longer term study, but hopefully that will throw up some interesting changes in why people perhaps progress more slowly beyond the different perhaps that's within even ALS. Okay, I think we're getting towards the end of it now, but I think at the end of our questions, I think we've got time for one more. A question here about some of the really emerging neurotechnology, brain computer interfaces. I'm not sure if anyone here has any insight into those. And as we're moving into this crazy technical world with, we already discussed AI, if anyone any insights onto where we're going next with brain computer interfaces and what do they promise?
So I might just, a thought that came to mind when I saw that question is a great idea. I mean, well, perhaps it would help function in the future, but what bothers me is that we still have to address pathology because that is an ongoing process in MND. And while you can retain function and improve some level of quality of life till you can address whatever is that process that is driving and spreading across the nervous system and primarily affecting the motor system. In MND, it does affect other regions of the brain, like the prefrontal cortex. So that's also an area to think about. So while, like brain computer interfaces or neural implants, I don't say why not. I mean, great idea, but somebody has to have the funding to see whether that could make a difference in MND. But what really bothers me is what about the disease which is still progressing? Can we turn that back by using neural interfaces, my thought.
And certainly, I think one of the, I think is a very clever form of brain-computer interfaces from an Australian, Thomas Oxley from University of Melbourne, has a company called Synchron, which is now based out of the US, and he's developed a brain-computer interface called Stentro. Rather than having to actually drill a hole and access the brain directly, they've inserted the computer interface into a stent that they can insert into the through your carotid artery, I think, and push it up over the top of you, so it can sit under your skull within a blood vessel. And they are trialing that, and they've already shown that patients using that can control a computer mouse just by thought. So, as you say, Parvathi, there's as an aid tool, it's great, but we're still nowhere near using this kind of approach as a therapy, I guess, which perhaps we can advance to that in the future. So I think we've reached the end of our time now. There's a few questions we didn't get to, but we can perhaps come to those in a later session, or perhaps even answer some of them offline after the meeting. So I'd just like to thank our fantastic panel. We've gone across a huge breadth of topics tonight, and you've done a fantastic job of really educating everyone, including myself as well. So for everyone watching tonight, everyone gets a link to a recording, so you can watch it again, and also send it around your own networks to make sure as many other people can get these fantastic insights as well. Just say a couple of events. We've got our research symposium happening in 2 or 3 weeks. It's open to everyone, people with lived experience as well, and it will be on Zoom as well for those of you who can't make it to Melbourne. And also just a reminder that if you are interested in contributing further to the MND community, we have now established the Lived Experience Network. There's a link up there in the chat. I think Laura put it up before. So you can sign up to that and apply your own lived experience to a whole host of issues, from research, advocacy, committees. We really want to have the lived experience community engaging right across the wider MND community. So again, thank you all tonight. I really appreciate the panelists, and we really appreciate everyone dialing in tonight. I think tonight was our biggest turnout ever, and thank you for the wide array of questions. So thank you all, and enjoy the rest of your evenings.